So thank you so much for inviting me. Mappings are really near and dear to my heart because they are foundational to helping the patients worldwide that we try to help in the rare disease space. So I wanted to give a keynote that really talked about sort of the implications of what happens with these mappings downstream in the real world. So the dangers of bad mappings, how imprecise and incomplete mappings can cost lives. And these slides are also available at that link, and I know we'll be posting them later, and feel free to tweet me at Antawaka. So in 1983 in the United States, we had something called the Orphan Drug Act. And in this act, it stated that nearly 1 out of every 10 Americans lives with at least 1 or more of 7,000 known rare diseases. And the reason that the Orphan Drug Act was created was because at that time, we had only 34 therapies for rare diseases. And now we have, actually it's probably around 900 different therapies for rare diseases. But the problem here isn't that we haven't made improvements in developing therapies, it's that we haven't changed that number of how many rare diseases there actually are since 1983. So if you look over there on the right, it still states on many websites, including this one on Global Genes, that there are 7,000 distinct rare diseases today. Well, we learn about new diseases every day, every week. So the number hasn't changed since 1983 because we aren't actually able to count them. And we aren't able to count them because we don't actually define them in the same way. So the criteria for what constitutes, why can't we count them? The criteria for what constitutes rare varies around the world. So in the US, the Orphan Drug Act defines a rare disease that affects fewer than 200,000 people. In 2000, the European Union considered a disease to be rare when it affects fewer than 1 in 2,000 people. So these different definitions in different parts of the world about what constitutes rare mean that we don't even define what a rare disease is in the same way worldwide. We know that new diseases are discovered all the time, but that number is never updated. So if you look at the Undiagnosed Disease Network or the literature or in the online Mendelian Inheritance in Man, we get new terms, new disease definitions all the time. We're constantly finding people who have an N of 1 or basically a disease that's never been identified before, and then finding that N of 2, another person in the world who might actually have the same genetic variant and therefore the same disease in systems like the GA4-GH matchmaker exchange. And so these disease definitions vary around the world. There are dozens of terminologies, as David Osumi-Sutherland mentioned earlier. And many different disease registries exist, but these are often not included in the clinical terminology such as ICD, which are commonly used in electronic health records. What that means is that if I go to my local clinical environment and I query our electronic health record for people that have Fanconi anemia, for example, which we have about a few thousand patients in the United States with that disease, I will find 0 results because there's no term for that disease. So we have no mechanism to actually even count the patients that have a rare disease very effectively. And so fundamentally, the definition of what a rare disease is and how to model it computationally has remained more of an art than a science. And so if we are able to, so it's not just that we don't know how many people have a rare disease or that we don't share the definitions, but because we can't count them, we also can't help diagnose them. We can't develop therapies for them. And so this fundamentally is a mapping problem. We have to map and align the information about rare diseases around the world. So why do I care about this? Yes, not being able to count the number of rare diseases means we don't have clear definitions. It makes it very hard to build diagnostic tools and reveal mechanisms. And yes, bad mappings can cost lives. So this is an example from the Genomics England 100,000 genomes program. And this is a little girl named Jessica. She was 4 when this was when her case first came to Genomics England. And she has a rare condition which causes epilepsy and affects her movement and developmental delay. All the standard genetics tests were negative for her. And I just wanted to bring a little context to our mapping problem here. So this child needs a diagnosis so we can understand how we can best care for her so that she can hopefully overcome her undiagnosed disease. But in order to diagnose her, we need to find and align all the available evidence that might help us understand what disease does she have so we can identify potential therapies or other care regimes. So the patient phenotyping is one of the first steps. So how do we actually describe her epilepsy or her developmental delays? That the reality is the way in which we do that isn't so different from this this old document written back in the day. The electronic health record is still full of notes that explain the characterization of clinically relevant phenotypes, but not necessarily those that are codable concepts that are used for billing, which is one of the main goals of the electronic health record. And so, we really, so this is much more than a mapping problem, it's a coding problem, but getting content out of free text and into some kind of coded terminology is one of the big challenges. So I wanted to talk a little bit about how we have done that using the human phenotype ontology, which is one of the flagship ontologies developed in the Monarch Initiative. And the way it works here is you basically you characterize your patient. So over on the left and on the right, we have two different patients, a little girl and a 14-year-old boy. And it turns out these two children actually have the same disease, Wiedemann-Steiner syndrome. And by creating a sort of knowledge representation of Wiedemann-Steiner syndrome that has the specific characteristics encoded using the human phenotype ontology, such as short toe, short middle phalanx of the finger, intellectual disability, microcephaly, we can actually use semantic similarity algorithms to match the patient's individual characteristics to that gold standard profile. And in this case, using the phenotypic characteristics coded in such a way, these two children are actually diagnosed in the same clinic within a few weeks of each other. It turns out they actually have the same disease, Wiedemann-Steiner syndrome, but they have different variants in the same gene, KMT2A. And then you can see some of the differences from the gold standard profile that they have. So the boy on the right has long toes, not short toes, and macrocephaly, not microcephaly. And so it's not necessarily, we're not looking for exact matches. We're looking for, overarching profile matches using the semantic similarity algorithms. And so this is the key task, is to diagnose patients using this phenotypic similarity method. So this is just to remind you that, so why is the human phenotype ontology such a fundamental resource for this process? Well, it's not just about having another terminology. It's that the terms within the human phenotype ontology, which are now around 15,000 terms, describe the phenotypic characterizations that one can have of an individual patient. And this is done in a way that treats the human patient much like an organism in terms of describing those characteristics. So you wouldn't necessarily have a billing code for something like hyposmia or the lack of sensory perception of smell, although nowadays with COVID, maybe you would. But for the most part, a lot of the terms in the human phenotype ontology are not really represented in clinical terminologies because they focus on these descriptors. But these are the very descriptors that allow us to integrate and compare with much other data that will help us diagnose patients like Jessica. So for example, that hyposmia term has an axiom that relates that term to the gene ontology sensory perception of smell. And when I made this slide some time ago, there was at the time 34,000 annotations from 22 different species. So there's all this data that's attached to the axioms that are available in how we define the term hyposmia in the human phenotype ontology. And it's that interoperability and those other data that can provide us the foundation to give us the evidence that we need for the diagnostics. So this is just some of the resource types that we pull into a big graph using ontologies. So we have many different data sources about genes, variants, models, diseases, phenotypes, many different data sources, many different ontologies, and then pulling those in these kind of Uber or sort of integrative resources such as Mondo, Eupheno, Uberon, and gene ontology, which really kind of helps us integrate across all these different resources. Well, these are the data that are then now available to provide evidence for inferring what disease, how we want to characterize the disease that a patient like Jessica might have. So we take those data and we combine it with genomic data, and we have a variant prioritization tool called Exomizer, which pulls in different data from different species, protein-protein interaction networks, and it combines those data with the genomic data to help identify which candidate variants might actually be the smoking gun that causes the disease in a patient like Jessica. And so it's that integrative data then that we really need to have access to in order to support the diagnosis. So the problem is that combining data that relates to different disease terminologies is very hard, and it's fundamental to the task at hand. So what is the most clinically useful way to define and group diseases? There are many, many different disease terminologies for different areas of clinical care. We have complex diseases, cancer, infectious diseases, rare diseases, Mendelian diseases, and there are dozens of disease ontologies. So they all have information that is relevant to our task at hand for diagnostics. But and so many of these terminologies already include mappings to each other. And we heard earlier from David Osumi Sutherland about some of the problems that we see with disease ontology mappings. These are often mutually inconsistent. There's n squared sets of mappings, and we don't understand what the one-to-one equivalents are. But if we think about the the little girl and the little boy that I showed earlier, if we don't actually understand what we're matching against, we can't perform that phenotypic similarity algorithm and thereby match the disease. So this is just an example. This was from a annual reviews article that we wrote that kind of talked about some of the problems with disease mapping. So we have many different terminologies. Some of them have text definitions, some of them are single hierarchies, some of them are multiple inheritance, some of them are more flat. Some of them have language translations, some of them have cross references, some of them have synonyms. And if you just look at examples here for like Ehlers-Danlos syndrome and pancreatic cancer, you see that the number of synonyms, the number of descendants, the number of parents is different in each one. So we have this incredible heterogeneity in the placement of the diseases hierarchically, and therefore their meaning, their mapping to each other, and the number and types of synonyms. And this is sort of the landscape on which, if we want to sort of figure out how many rare diseases there are and attach the appropriate phenotypic features to each one, and thereby use that information as the gold standard to match our patients, we need to fix this problem. So this is where, and we're going to hear from Jim Bellhoff about K-Boom and Boomer tomorrow. But essentially, we created Mondo to reconcile all the different disease ontologies using Chris Mungel's original algorithm, K-Boom, which is the Bayesian owl ontology merging algorithm, which combines logical and probabilistic inference to create a merged graph of the different relevant terminologies that you see on the left. And we have many more now aligned. That ontology then is curated for looking for inconsistencies across the different terminologies, and David showed some nice examples this morning. So those then get curated in terms of their equivalence relationships, so that we can create merged cliques of the terms from the different ontologies and really align the knowledge that we get from all those different disease ontologies, so we can have a handle on which to hang the phenotypic features that we're going to use as the comparator for the diagnostic tools. So when we took that, so a little more recently, and there's a link there at the bottom, we actually looked at just 5 sources for where we took them in the merged ontology that is Mondo. 5 sources comprised 10,577 unique rare disease concepts, which is a lot more than the 7,000 or 7,500 that we're still posting on every website about how many rare diseases there are in the world. And so that's quite substantial. And in fact, other estimates from other projects have suggested that we may even have as many as 12,000 rare diseases to date. So we're starting to get a little closer to understanding even just how to count them. But when you look across all the different resources, many of the resources are only in one source. So here we have a lot of diseases, 1,600 of them that are only in OrphaNet and not in any of the other 4 resources, the NCIT, the Disease Ontology, GARD, or OMIM. Only 333 disease concepts were in all 5 sources after this equivalence merging technique was performed. This is astonishingly low number. And when you think about trying to align all these different ontologies so that we can actually have those handles for that diagnostic task, it is alarming. If you are using diagnostic tools that do not use all of the resources, you have a much lower chance of getting a diagnosis. So we need the knowledge from all of these different resources to be merged together into one unified framework so that we can actually use that knowledge to help diagnose rare disease patients. So mapping itself is not going to solve this problem. It's what we attach to these diseases that actually matters. So Lynn actually talked a little bit about the lumping and splitting problem this morning as well. So defining diseases is actually really challenging. And this is a project that we have between ClinGen and Monarch, which is called the Lumping and Splitting Committee and Disease Naming Committee, where we actually talk about what are the sort of rules of the road for how we define a new disease. How do we get, the 12,001st disease? Does it have a distinct molecular mechanism? Is there a reputable assertion or difference between the diseases? Does it have different clinical management? Does it have distinct phenotypic profiles? So these are all the different kinds of things to consider when you think about creating a new disease entity and a new identifier and the new information that's actually associated with that. And so really each disease identifier is associated with a little model of disease. So if the disease identifier or Mondo ID is in the middle, we have a bunch of different information that's associated with that disease that forms the framework by which we want to compare those patients like Jessica. So when is the disease or phenotype onset? What genes or variants are associated with that disease? What's the heritability? What treatments are available? Are there environmental factors that are either causal or influential on the disease? And so all of this information forms a little disease model that can then be used as the basis for comparison. So this is just an example from Mondo of dead simple ontology design patterns, which we'll hear more about as well. DOS-DPs, which are pre-made patterns that specify the label, the text definition, and synonyms, but specifically the logical definition based on these disease attributes. So in this case, we're looking at diseases that are realized in response to an exposure. So these patterns, examples have chemically induced disorders such as alcohol amnestic disorder. And you can see the sort of exposure events and the sort of the little YAML file that explains the pattern that is used to define this disease set of classes. And so what that means is that all of the different diseases that fall under this parent term within Mondo would be automatically generated according to this pattern, with the different slots having different types of exposure events. And so this allows us to really align and encode in a really consistent manner all of the different disease classes from all the different resources. So next, I want to talk a little bit about individuals. How do we represent that information about an individual? So we've been working in the Global Alliance for Genomics and Health to develop what is called phenopackets. And phenopackets is essentially a little schema that you can write about a patient and their characteristics. Do they have what phenotypes are observed or not observed? How are they linked to the patient? How are they linked to the genomic information, to samples, to parents and siblings? How severe are they? When were they first observed? And this just forms essentially an individual case level bit of information that we can use to compare patients against our gold standard definitions that I talked about earlier. So here we have, for example, the model of the disease over on the right that I talked about. That's our gold standard reference. And an individual patient over on the left that has a phenopacket that has been written about them. In this case, we have a 14-year-old boy. They're male. They have a peripheral neuropathy. And in this case, we have a reference for their disease that has a COL6A1 mutation that leads to Bethlem myopathy with recurrent hematuria. And so in this case, what we want to try to do, and this is a different kind of mapping problem, because what we're really trying to do is say, if we had a world full of shared phenopackets about individual cases, we would be able to actually better match these cases to these reference knowledge artifacts that we have in our models in Mondo. But the problem is that it's very hard to actually get information about individuals. And so we actually often don't really know what is the variability in certain kinds of things. When does peripheral neuropathy start for patients that have Bethlem myopathy? And so moving away from just summary level information in manuscripts to case level information that is shared more broadly will allow us to derive the models that we see on the right instead of and iterate between individual level information and these generalized models that we used to compare for the diagnostic use. So this is a different kind of quote mapping problem where we have to understand the relationship between individuals with a rare disease and the generalized reference models that we're using to help with the diagnosis. So each standardized disease has a different phenotype profile. So this just kind of gives you a sense. So for example, if we have an abnormality of the orbital region over here, that's a human phenotype ontology. There's 2,644 diseases that are associated with that phenotypic term. But as you get to more specific phenotypic terms like hypertelorism, we have 94 diseases that are associated with that term. And so you can just see how the relationship between the phenotypic profiles and the Mondo terms functions in terms of those gold standard profiles. So the different communities also annotate different relationships, and this makes determining the equivalency between the terms really challenging. So if we think about the model that I showed with the different information, variants, genes, phenotypes, treatments, and exposures, different resources capture different curated elements of this information. So ClinVar captures diseases to variants. OMIM captures diseases to genes. The Monarch Initiative captures diseases to phenotypes, and so on. And so nobody actually curates the entire model, but the entire model is what we need as that associated information to have our gold standards for our diagnostic tools. So if we could actually get everyone to agree on the same overarching model, then we would and divide and conquer how we curate this information, we would have a much better chance of defining more accurately and more consistently around the globe all of those diseases that are now more than 10,000. So map utility is in the eye of the beholder. And so this is really what we're facing in terms of how we think about mapping in the rare disease space. So the takeaways are that we need axiom and graph alignment to define rare diseases in the same way around the world. We need to model disease attributes in the same way around the world. And we need to model individuals and share this information around the world to be able to compare them against standard disease definitions. These are all different kinds of mapping challenges. But essentially, if we achieve these three mapping challenges and we overcome these mapping challenges, that will result in saving lives and being able to diagnose patients and identify their treatments that are needed. So just wanted to make a call for participation for anyone who's interested in helping with Mondo. There's a lot of terrific information here. And I also wanted to thank in particular the NHGRI for funding our Phenomics First resource, which has really helped bring the community together. And we really, really welcome anybody's participation. You can find out more at mondo.monarchinitiative.org. I also want to thank all the users and contributors. We have a huge number of resources working together to help align the content from all their different contexts. And so it's their usage that actually has allowed us to really build out Mondo for those diagnostic use cases. And then, of course, all of the individuals, but in particular, Chris Mongol, who really launched a Mondo with KBOOM, and Nicole Vasilevsky, who's been our lead curator all along, and Miko, who will be talking a little bit more about SSSOM, who's also really helped design a lot of the patterns and the ways in which we encode our mapping file. And so with that, I would be happy to take some questions.
That was a fantastic talk. I really enjoyed that. So yes, audience members, please put questions in the chat, and while those drive in, I will kick it off. So I really like this idea of creating those pheno packets for patients and clinical data. But who, in your mind, who's the onus on to create those pheno packets? Is it the clinician? Is it the physician? Is it the lab scientist at the bench? Where does it start?
So that's a very good question, and we've really designed phenopackets to date to really be useful and generatable in a lot of different contexts. So, for one, just to start, journal articles. In journal articles, we often see summary level tables that essentially explain how many patients have X phenotypic feature, but it doesn't say which phenotypic features each patient has. So there are these tables of summary information, but we actually need to understand the sort of co-occurrence of phenotypic features and the variation of the co-occurrence in order to understand how to create, how to lump or split the different disease definitions. And so we really hope that journals will start requiring phenopackets for these. These are often small cohorts of patients with a given rare disease, instead of these summary level tables. So that's one place where we can have a huge difference. And then in terms of clinically, it's quite straightforward for a clinician, like clinical geneticist or genomic medicine physician, to create a phenopacket for their patients and be able to share that information. The patients themselves often want that information shared, and it's not really so different from what they're already sharing on places like Facebook or Patients Like Me. And so, I think those are contexts as well where patients can work together with their clinicians to generate them. I didn't talk about it today, but we also have generated a layperson translation of the human phenotype ontology. And that layperson translation turns out to be it covers about a third of the ontology, where some of the terms are just too clinical to actually, translate into layperson ease. But for those terms that are available to the patient, they're actually quite robust when we do our analyses to see how diagnostically useful they are. And actually the sort of union of patient-generated terms with clinically generated terms is our ideal, because oftentimes patients generate information about themselves that you wouldn't otherwise observe in the clinic. So for example, a baby that's inconsolable or doesn't have any tears is not necessarily something you would notice when the baby is in the clinic. And so that information combined with the clinical information is sort of maximizing the characterization. So patients themselves can also generate phenopackets and share them in those contexts as well. So we also have finally a project to generate phenopackets directly out of the electronic health record, but that turns out to be very challenging for the reasons I mentioned at the beginning, because the information that we need for diagnostics is scattered throughout the EHR. A lot of it's in the notes or in the annotations on a pathology report. There's some structured data in the labs or the problem list, but it all has to sort of be collated and encoded in the same structured way, and then exported. And so we're using FHIR to actually create phenopackets exports from EHRs, but it's also up to the clinicians to document and sort of check that information, because it is sort of scattered around the EHR. The EHR is not designed for phenotyping. So.
Yeah, and I know we have some attendees from Elsevier, so hopefully the journal editors are listening in and can follow up later. A few questions in the chat. So I see one from Philip Patton asking, how do you see phenopackets compared to OMOP-CDM? Is there any interest to let the two initiatives collaborate?
Yeah, that's a terrific question. So I'm quite familiar with OMOP, and we've been actually working to put Mondo and HPO into OMOP as terminologies that are available to OMOP, and similarly also in HL7 for FHIR, so that so clinicians can actually start, basically like the Fanconi anemia example, that Fanconi anemia will start being available in more broadly in clinical context. For OMOP, OMOP is, I think that there could actually, similar to what we're doing with FHIR, and maybe it's actually easier with OMOP, we could actually create a phenopacket derivative from OMOP quite readily, I would think. There's the content fields within OMOP are actually much more aligned, because they're designed for research. They're much more aligned with the use case of phenopackets for diagnostics, whereas the content in an EHR directly is not really designed for research, right? And so I think it actually would be quite straightforward to do the alignment with OMOP, especially once we have the concept set alignment finished with Mondo and HPO. So if you're interested in that, please contact me. I'd be delighted to talk to you about how we might approach that.
Great. And I just see the questions keep rolling in. So from Martin, would clinical case report publications be a data source for extracting phenotypic packets or validate that they could be extracted or are covered by existing ontologies? Are there negated instances for some concepts of clinical relevance?
Absolutely. So one of the things that we've been doing is actually Peter Robinson's been leading the development of some tooling that helps take case reports and text mines them and curates them for phenotypic packets and generates phenotypic packets. Partly, and we do have some journals that have been interested in kind of trying this out with us. But that is our hope, is in fact that case reports and these other sort of small rare disease cohort manuscripts will be a source of phenotypic packets. And again, it's really getting after this tension between how we describe individuals and how do we summarize that information into these knowledge artifacts that surround a Mondo disease identifier that we then use as the comparator for the diagnostics. And there's a virtuous cycle in an ideal world where if we had phenotypic packets about every person, and every person really has their own disease, we would be able to derive the useful classifications from that, and those would then become the disease entities over time. Obviously, that's very idealized and probably not ever going to happen. But I think a good start is really focusing on the literature, where just a more complete characterization and more computationally accessible set of phenotypic information about each individual that is presented in a case report or in these small cohort studies would be enormously helpful to the computational community.
Great. I see a question from Haendel on the chat. It's a bit open-ended, but she's hoping that you can elaborate on the idea of using the axioms for mapping and aligning and modeling efforts.
Yeah, so we're going to hear a little bit more about that from Nico tomorrow when he talks about SSSOM. But essentially, when we think about how we do the alignment, we need to take into account many different attributes of the ontologies. And we heard a little bit about that this morning from the Ontology Alignment Initiative as well. So there's synonyms, there's cross references, there's the overall hierarchy. But if you actually leverage the axioms themselves, and the DOS-DP patterns, you can actually identify where the inconsistencies are. And we often find cycles or things where, ontologies that are mapped to each other will map a parent and child in the opposite direction, so that you have a parent and a child in one ontology, but they're flipped in the other ontology. The axioms actually sort of expose a lot of these types of inconsistencies when you're trying to align the different ontologies. And so that logical inference really helps us to understand what can we actually define as an equivalent set of clique of different terms from different ontologies. So it's really sort of trying to reveal the quote real ontology that exists from all the different resources based upon that underlying logic.
Well, that's really awesome. Yeah, I mean, adding axioms to ontologies can save lives, maybe. I really encourage all the ontology builders on this call to add more axioms. I think that's fantastic that you guys were working on that. Very nice.
Thank you. Thank you very much.
And I think we'll just end this section with, this is a big effort. And so I see a question for Dermot. Can you just restate the key needs for creating such an ambitious and comprehensive graph?
I think that there are many different goals. One goal is simply to align our knowledge. So when you think about the rare disease use case, one of the challenges that we have is that we just don't have very much information about any of these rare diseases. So it might be that a patient has a rare disease, and we don't actually know what the molecular mechanism is, even what's the smoking gun gene. So how can we compare that the phenotypic features of that patient against other organisms where we know a lot more potentially about the candidate genes that might be causal for that disease. So we need this ability to integrate data from other organisms, from other types of resources, protein-protein interaction, transcription data, things that can kind of help fill in the gaps where we actually just don't know very much in humans. Humans turns out to be very poor experimental subjects, so we don't actually have a lot of data on a lot of the genes in humans. And what we derive, we often have to derive from things like cell lines. And so it's really important that we have the ability to integrate data from a lot of different resources to bring knowledge to these problems with rare diseases. So that's one thing. And that's really thinking about not just diagnostics, but also about mechanism discovery so that we can identify therapies. So one of the biggest challenges in rare disease research is, how do you design a therapy when you only have, a few patients worldwide? With the new technologies such as RNA drugs, we have the opportunity to really develop precision therapies, but we have to understand the underlying mechanisms and understand what to target. And so the knowledge graphs can really help bring that, enhanced knowledge that can then reveal potential targets for drug development. I think it's also important for predictive analytics as well. So for rare disease, where we only have a few patients worldwide, we want to try to be predictive about what the outcomes might be, so that we can get ahead of them and hopefully have a better care regime for those patients as well. So I'd say those are some of the main goals that we have. So diagnostics, treatment, development, and mechanism, understanding, and predictive analytics.